So at the beginning, when we started doing next generation sequencing for PGS, for aneuploidy screening, everything was wonderful. I mean, we were able to analyze the embryos to determine if there are normal, or not, and we were able to recommend their implantation or not according to the result. But after working a while, we found some surprises. The first one is that thanks to the high sensitivity of next-generation sequencing, now we know that some embryos are mosaic. That means that they are a mix between normal and abnormal cells. With microarrays, those embryos were classified as normal or abnormal depending on the place you did the biopsy. And the mosaic embryos have an amazing characteristic. That is that sometimes they are able to implant and to give birth a healthy baby. So now with next generation sequencing, we are able to classify the embryos in three categories: euploid, aneuploid, and mosaic embryos. And we can prioritize, obviously, the euploid embryos, and then the mosaic, just if everything fails, then we can try with the mosaic embryos. The other amazing thing was that now we are able to study the mitochondria level in the embryos, because now we know that when an embryo has some problems to develop, he's stressed, he's struggling to survive, and the way in which he can try to survive is producing more energy, and he do this producing more mitochondria. So we can have the ratio between the mitochondrial DNA and the nuclear DNA, and to have an estimation if this embryo is stressed or not. So now, for the question if an embryo can be transferred or not according to the results, we have more answers. So that was for PGS, for aneuploid screening. As I said, we can do also PGD for monogenic disorders. With next-generation sequencing, we can do very nice things. For example, we can do PGS and PGD at the same time. Think that before the next-generation sequencing, it was very difficult to combine PGS and PGD, because one was done with microarrays and the other with Sanger sequencing. But now it's quite easy to combine both. And who knows, maybe in the future, this will be the normal way to have babies. So thank you very much.
Fascinating talk. Thank you.
Thank you.
All right, questions? So, yes, was bound to come given your CRISPR conversations. What point is genomics going to change the ethics of PGS, PGD, and embryo editing?
Very difficult question to be the first one. I mean, I don't have an answer, because what is ethics or not depends on the society and on the people. The key point here is it is legal or not to do these kind of things, and it is in many countries. So the problem here is that if people think that they need to do genome editing, they will travel whenever to do it.
Sure. Okay, regarding embryo phenotype prediction, what about the environmental effects, and does it make sense to talk about genetic determinism for all traits? And I'd like to link it back to the previous speaker as well, right? With the microbiome having such a huge impact on people's health and lives, how do you balance these two? Because what you talked about with human longevity was well, I'll take a look at your germline and try to predict things, as opposed to all of the other epigenetic and microbiotic factors.
Yeah. It's true that there is no, the prediction is not exact. There is a lot of variation. So if you look at the picture in detail, you can see a lot of difference. And those differences are coming mainly from epigenomics. So it's just a prediction, just an approximation.
Okay. So in your mind, it doesn't make sense to speak from there, just from all traits. Okay. Actually, this is a good one. So why NGS? And you started going into some of these, right? But what's the main driver between everything moving to NGS at some point, and as opposed to arrays, which can also detect SNPs, all kinds?
I think we are moving to NGS just for an economical reason, because, as I said, the IVF cycle is quite expensive, and genetics is also quite expensive. If we do microarrays and Sanger sequencing and very different techniques with, as I said, with a lot of people working at the same time. So the reason to move to NGS is just because it is cheaper. And regarding to aneuploidy study, we have even better resolution because now we can identify mosaicism.
And is that the same trend you're seeing even in carrier screening? So do you think carrier screening will move eventually all to NGS, or is that still within the domain of microarrays?
Yeah, I think carrier screening is moving to NGS. But the problem moving carrier screening into NGS is that we will have a lot of variants of uncertain significant. So we will not be able to give recommendations to the couple to go to an IVF cycle or not. So
interesting. Even if you use panels? Even with panels, you're finding a lot of...
Yeah.
Okay. Time for maybe one more question. So what further advances in NGS technology would you like to see from your perspective and what you're doing in terms of reproductive health?
Well, from the technical point of view, what I would like to see is first to be able to sequence the embryo and to perform the analysis without the amplification, because the amplification gives us a lot of errors and uncertainties. So I would like to see doing this without amplification, and we'll be able to sequence the whole embryo to see the novel mutations.
Would molecular tagging help at all with that?
Yeah, maybe.
Perhaps, yeah. Okay. Any questions from the audience? There we go. Francisco? Oh, nice.
All right, so I was asking Geeta to pose this for me, but since you said this is the last question, how soon into the future do you see CRISPR in regular clinical practice?
Yeah, I think CRISPR is something that we have to do. It will be for sure something normal, something current. I mean, in reproduction, it will be adopted by a lot of people for sure.
But how soon? Are we talking, 2 years, 5 years, 10 years?
I don't know, but I think in 2 years maybe. Yeah, okay. Because there is some groups doing very nice research on this.
Okay, thank you.
Welcome.
Well, thank you, Dr. Alcaraz.
Thank you.
Fascinating talk. Yeah, thanks again.